Hello everyone, welcome to my Study YouTube channel. Today I am going to demonstrate cd-hit tool to identify non-redundant data from the FASTA file. So first we will go to the theory part. So cd-hit actually it detect the cluster database. So it identify the high identity tolerance and make a non-redundant file. So to do it, go to the Google and type cd-hit download and go to this GitHub page and download this source code zip file. So download is complete. Let's check it. So here is the cd-hit file. So I have one redundant FASTA file. I made this file from the Trinity de novo transcriptome analysis of Saccharomyces cerevisiae from this file, and so it is a representative of the FASTA file. Here is the three sequence 1, that is sequence 2, and this is sequence 3. So maybe these files are redundant, maybe one sequence representing the other sequence as a homology. So to check it, just extract it first. So extraction is done. Okay, let's open a terminal here and check the list. So to install it, you have to just write one command, make and enter. So I guess it is done. Let's check the list again. So here you can see that this is the cd-hit and cd-hit-est is in executable format. So to check it, the /, then come to cd-hit, then hyphen est, est, and enter. So here you can see the full usage of this tool. So let's clear it. So I'm going to put this redundant file. Let's copy it and paste it here. So I'm going to convert this redundant FASTA file into the non-redundant FASTA file. For that, let's check it. So here is my redundant FASTA file, and let's again /cd hit -est and -i for input. Here is the input file is Redundant.fasta, and I have to -o for output. I am going to name it non-redundant. So it is the total script. Let's run it. So it is a very short read of 3 sequence. Let's check the output. So here is the output, not redundant. Let's check it. So here is the one file, FASTA file, and there's the second FASTA file. So I gave 3 FASTA file in input, and here is the 2 FASTA file. It means just one sequence is representing the 2 sequence of the input file. Let's check the non-redundant cluster. So here is the, you can see here is the 2 cluster 0 and cluster 1. In cluster 0 there is only one representative FASTA file, and the cluster 2 here is a 2 representative FASTA file. So it is the clustering to make the non-redundant database. So it is all about the cd-hit. If you found this tutorial helpful, please give it a like and leave a comment. For more bioinformatics tutorial, don't forget to subscribe to my channel. Thank you.